for all of these trials and can prove what we think we are actually proving. Thank you, Lisa. Excuse me. Lisa from the NCI here, of course.
Okay, so it's always a daunting task to try and come up with what to say about statistical considerations when the topics covered are so broad. So I've picked these to find a couple of issues that I see often in biomarker studies, and particularly as we're trying to advance biomarker assays to something that's actually a clinically useful tool. So you'll have to forgive me if I fly through some of the slides. I am going to make them available, so you will be able to take a look at anything that you missed. And I also will admit that many of my examples are, in fact, most of my examples are not cell-free DNA assays because the issues really apply more broadly. That's the nice thing about being a statistician. Usually your points can apply to different topics. So I always like to start off reminding people that there are, in fact, many different uses of biomarker-based tests. And for the purposes of this talk, I'm going to focus more on the three boxes to the left. So these are patients who have already been diagnosed with cancer, and we're going to follow them through their workup for looking at prognostic and predictive markers and also doing some monitoring during and after treatment. To remind everyone of different meaning for validation, I always like to point out that people throw that term into a paper thinking they have proven something, but it's not always clear what they've proven. And I think it's helpful to break it into the three categories I have listed here. Analytical validation, which is just basically how well is the test doing what it's supposed to do technically, how well you're measuring the biomarker. Clinical validation is what we see in most published papers where somebody's taken a biomarker and they've shown that it associates with some kind of clinical endpoint. That may or may not actually be useful, but it gets you a significant p-value in a publication. And we have a lot of trouble going from that clinical validation to something that's actually clinically useful, which is what I mean by clinical utility. So ideally, clinical utility means that use of this biomarker test will actually lead to some kind of benefit from the patient. It steers them to a better treatment. It gives information in some way that a standard test could give, but it does it more cheaply, less invasively, and just basically easier, better way to do it. This is just a 10-second commercial. Since I am going to be giving some definitions here, I want to make you aware of a glossary that's been built by a joint NIH-FDA committee to really try and refine some of the definitions associated with biomarkers and endpoints. We've decided that in order to have an intelligent discussion about levels of evidence required, we have to all be using the same vocabulary. So that was really the purpose of that glossary. So I'll start with the first class of biomarker, prognostic biomarker, and I think everyone knows what that means. Simply, it tells you something about patient's clinical course. Are they going to recur, die, or respond to have a good response? But this is where we often get hung up in biomarker papers, where we show an association with an outcome like, for example, overall survival. And we look at those two sets of curves that I've given you and say, well, how would I use that clinically? So left, you might say, well, if I can identify patients who either without additional therapy or perhaps on a standard therapy very, very well by biomarker, so the blue dashed line in the left plot, that might be useful because then I can not have to give that patient more toxic therapy. On the right, though, many of the survival curves you'll see in papers look more like that, where you see a separation, probably statistically significant, but does it actually help me in any way with how I would manage this patient? It may or may not. Often it doesn't, unless one of those groups is associated with the biomarker that we can target. But that really gets into another class of biomarkers. So some challenges in interpreting the literature or things to think about as you're doing your own biomarker studies as they relate to prognostic biomarkers. Cut point optimization is a big issue to deal with. The composition of the patient cohorts may vary from study to study, and absolute risk estimates often are not well described. People tend to just report things like hazard ratio. and I'm going to explain these further in the next couple of slides. This was an example. This was a protein marker. It was L-asparaginase protein levels. There was an initial discovery cohort, and they found that if they optimized the cut point at 75% stained cells, that they could get a nice survival separation. And that looks kind of like that earlier plot that I said, well, that might actually be clinically useful because that good risk group has very good outcome, and that might tell you something about the need or not for the therapy. When the same assay and cut point were applied to a validation cohort, which is the middle plot, you can see things have changed. So the result was still statistically significant, but the hazard ratio now has been basically cut by more than half. And that upper curve, the good risk curve, is not quite as good as it used to be. Okay, so have we established clinical in this case? Probably not. You know, we need to understand more about where we had all these shifts of the curve. Another thing that could have played a role here is that the discovery cohort and the validation cohort had different distribution of standard prognostic variables. So you can see there's a shift in grade distribution, for example, going from the discovery cohort to the validation cohort. That may have had something to do with the curves shifting down in the validation cohort. So you have to really keep track of all these things when you're trying to do validation or evaluate somebody else's claim of a validation. There's a lot of moving parts. You'd be thinking about this early in the process if you really want to move your biomarker to something that's clinically useful. Another thing that gets sort of tripped up is the difference between a relative risk and an absolute risk. So again, the plot on the left we think could potentially be clinically useful because that upper curve looks so good. It's really high. But if you looked at hazard ratios, that wouldn't be so obvious. And also if you looked at the difference, the absolute difference in the curves, you can see that on the left you have a distance D1 and on the right you have a distance D2. So actually the absolute difference between the curves is bigger on the right than it is on the left, yet probably the left would be more clinically useful. Here's another type of prognostic biomarker, but really measured after some therapy has been given. This was an example that occurred in getting ready for a trial on nasopharyngeal carcinoma. They found that there were plenty of data supporting the notion that plasma Epstein-Barr virus DNA was a good prognostic factor if measured after some initial course of therapy. So this was pretty good, and of course the treatment for nasopharyngeal carcinoma is pretty awful. So if you could find a good risk group where you might be able to either back off treatment or you might be able to say, well, we're not going to subject these patients to any more treatment, and then your worst risk group, that low curve, maybe you could give some strong treatment or additional treatment. The wrong way. This was the study design that was going to try and exploit that prognostic association, that people would go through their initial course of therapy and they would have the plasma EBV DNA measurement made. If there was still detectable EBV DNA, they would go into one randomization, which is a randomization between control, which is a standard therapy, adjuvant therapy, versus an experimental arm that could potentially give these patients a better outcome. And then if you were low risk, meaning you had undetectable plasma EBV, then you would be randomized to control versus observation. If that worked, and the trial is still ongoing, if that worked, you would be sparing some patients some therapy, and you would be giving additional therapy that hopefully would improve the outcome for the patients who had the poor prognosis. But as part of getting ready for the trial, this is an aspect people often forget, that you really need to make sure you understand the analytical performance of your assay. So the investigators were very thoughtful and had done a study to see if the labs they would be using for the trial concordant results. So there were 40 samples sent around, and there were 4 different labs. This was going to be an international study. In Taiwan, 1 in Hong Kong, and then Stanford was sort of the reference lab in the U.S. And much everyone's surprised, I think, of the 40 samples that they sent around, the EBV detection rates, which again was going to determine which of those parts of the trial you ended up in, differed dramatically. Okay? So it's a good thing they did that, because you can imagine the results of this trial would be totally uninterpretable if it depended which lab your sample was sent to. You'd be thoroughly mixing these arms. So what they did is they did a harmonization effort, found out that they had to harmonize PCR master mixes and calibrators. After they did all that, they substantially improved the concordance across the laboratories. The ICC that you see there, that's a measurement that really just explains the proportion of total variation that is explained by the between-subject variability relative to the full variability, which would be the between patient and the sort of technical variability in the assay. So an ICC 1 would be perfect reproducibility. So you can see we went from things in the 60-ish to 70 range for the ICC up to things that were in mid-80s to better. This was definitely an important step to take, and one, again, that people don't think about, or they don't allow enough time to work out these assay details before they're ready to launch the study. We have many cases we get a clinical trial protocol that has an assay embedded in it, and it's like, oh, has anyone done these studies on the technical performance of the assay? And the answer is often no. And then so why scramble to try and come up with the resources and do this to not delay the trial? Predictive marker is another type of marker that I will touch on. Predictive basically means that you can use it to make decisions about treatment. So the ideal situation would be that you measure the marker, and if you're marker positive, it says you're going to do better on this treatment A. If you're marker negative, you'll do better on treatment B. Although we've been a little sloppy about how we use the term predictive, and I've seen it used in about 3 different ways, which I will explain. They are all predictive in a sense, but the kind of evidence you have to come up with to establish that something is in one of those 3 classes of predictive markers is a little different. So selection biomarker is what I just described, that you measure the marker, it says, okay, based on the marker, it tells me to give you treatment A or treatment B. That's what I'm calling treatment selection biomarker. Enrichment predictive biomarkers, what we actually have a lot of, and especially as we're developing targeted therapies, we say, well, we know based on the biology in our preclinical studies that we don't think this EGFR inhibitor is going to work in patients who don't have EGFR mutation. Okay, so we often will do our trials as what's called enrichment trials. So we use as an eligibility screen the biomarker, and then we do our usual randomization of treatment to targeted versus whatever the standard is, and then we can make a statement at the end of the trial about whether or not the new treatment is better than the alternative in the biomarker positive patients. What doesn't give us is information about patients who do not have that biomarker. And frankly, most of our predictive biomarkers are what I refer to as sloppy biomarkers. We sort of think we know the driver of the biology, but there's probably a lot of other stuff going on, and we're not getting it exactly right, and probably shouldn't expect we ever will get it exactly right. But just something to keep in mind. So the evidence you're going to collect is different depending on how you run the trial. In early phase trials, we often use the term predictive as something that will tell us the likelihood that the patient's tumor will respond to the therapy. So a couple of things to keep in mind there, of course. Response does not always correspond to a survival benefit. And even if you have 2 different agents which both will produce tumor responses, just because the tumor response rate on treatment A is better than the tumor response rate on treatment B does not necessarily mean that it translates in that same order to a survival endpoint. Okay. Another confusion we see often is the confusion between prognostic and predictive. So I often see plots like you see in the upper left there where everybody gets the new treatment, you measure the marker, say, hey, I have found a predictive marker for this treatment, namely if you're marker positive, you're going to do better on the new treatment than if you're marker negative. So one would think that means you should measure the marker and maybe give the new treatment to only the marker positives until you take a look at what happens under a standard therapy, which is what you see in the upper right. So if I draw in the curves for a standard therapy, we see that, in fact, even under the standard therapy, the marker positives do better than the marker negatives. Okay? So this is a confusion between prognostic and predictive. The marker on the top row there, that will not tell you a darn thing about who should get the treatment. It simply tells you that all these who are marker positive are going to do better than the ones who are marker negative. But this can actually happen in the reverse direction as well, where you treat everybody and you see that the curves are right on top of one another. You say, oh, defeat, can't publish this until, again, you bring in the standard curves and you see there that, in fact, something very important was going on, that the patients who were marker positive actually had their survival curve bumped up and made them have survival equivalent to the ones who were marker negative on the standard therapy. Okay, so the ideal kind of picture that we would like to see is what you see in this plot, where for the biomarker positives, you get this nice separation, meaning that the positive benefit from the new treatment. And if you're biomarker negative, either the curves are on top of one another, as you see in the lower right, or there's some separation but going in the different direction. So this is a really clear-cut message that this marker would be useful in selecting therapy. But often I see differences. What I see are split up kind of the other way, where you say, okay, for the new treatment, what's the difference in the survival for the different biomarker groups, and for the standard therapy, what's the difference? Okay, well here you might say, well, hey, this looks like this will be a useful marker for selecting who should get the new therapy, but not so fast, as if you put those curves together, what you actually see is that both groups, the marker positives and the marker negatives, benefit from the new therapy just by different amounts, which is how you got that picture on the last slide. So statistical language for what I just described refers to what we call a treatment by biomarker interaction, when you have treatment benefiting everyone, but maybe by different amounts, depending on the marker status. That's what we'd call in statistics a quantitative interaction. The other way, where you get this actual reversal, or perhaps you get nothing in the one group and you get a benefit in the other, that's called a qualitative interaction. So just a couple of real examples. This was a plasma biomarker, plasma IL-6, in hepatic renal cell cancer. And here someone was on sort of autopilot, and they did their statistical test for the treatment by biomarker interaction, which is, how we test this. And they got a significant interaction. But if you look at the plots the right way, which I've shown in the bottom row, you see that if you have high IL-6, you benefit from pazopanib. If you have low IL-6, you also benefit from pazopanib. So this is not going to tell me who I should give pazopanib to. Now, why does it happen like this? Maybe we just got the biomarker wrong. Maybe we got the biomarker cut point wrong. So we've kind of muddied the boundaries between those who benefit and don't benefit. We really can't tell. But just, a word of caution, you have to look at your data. Don't just, run things through a statistical program and look at p-values. Now, here's an example that has sort of the classic predictive biomarker that's really useful. That one of the classic papers on the EGFR mutation in this case, it was gefitinib that was being evaluated against carboplatin plus paclitaxel. You can see that you get this funny-looking survival picture. If you lump all the patients together, the survival curves actually cross. If you split them out into EGFR mutation positive versus negative, you see what we all now know, that if you're EGFR mutation positive, you tend to benefit from the targeted agent, whereas if you're negative, you won't. That was on progression-free survival. Things, of course, can get a little more complicated as you move to overall survival as the endpoint. So you have to keep track of your endpoints, too. Something that might be predictive for one endpoint may not be for another. Of course, in this particular thing of advanced lung cancer, with we're giving chemo versus targeted agent, there are a lot of such therapies these patients could get. A lot can happen from the time that the patient progresses to they reach their overall survival endpoint. Another thing that plagues us when we try and do things retrospectively like this is that we may not have the specimens available. Even if we have specimens available, a certain percentage will not actually be usable for running the assay. So lots of things that can start muddying the picture. And when they eventually did publish the overall survival results, you can see that, in fact, you looked at the upper right there is all the patients together. And the intent to treat population, plot B is where you have the EGFR mutation positive. Surprisingly, no separation in the survival curves. Looked at C was the mutation negative, no separation in the survival curves. The only place, and it's not large in absolute amount, the only place where they saw significant benefit was the patients who didn't have an EGFR result. Okay, so that's kind of interesting. However, there's probably an explanation for that. There are a couple of explanations. One is this was an Asian population, so of the ones who didn't benefit, probably a large portion were in fact EGFR mutation positive. Also, there were high rates of crossover to other treatments, many of those treatments being EGFR inhibitors. So you can see how the picture gets very easily muddied. Some clinical trial designs for getting predictive markers. I'm going to kind of speed through this because I've already touched on it. The basic 3 strategies we use are the enrichment design, which I already mentioned, where you use marker positivity as an eligibility criterion, you randomize, and you either see a benefit or you don't. And the obvious disadvantage of that is you don't get information on the marker negatives, and you can't go back later and refine the biomarker. Biomarker strategy design is something that sounds very attractive to people, which is that you randomize people to measuring the marker or not measuring the marker. And for those who get randomized to measuring the marker, you then have treatment determined by the marker value. Okay, so it sounds very intuitive, natural, seems like the way we ought to do all these things. Statisticians hate this design. Why do we hate it? Because if you are marker negative, it doesn't matter which of the arms you were randomized to, you will get the same treatment. So you're wasting all that information from the biomarker negative patients. Okay, that's where the statistical inefficiency comes from. Biomarker stratified design is the one we like to do most, where you get information on everybody and you've randomized everybody, assuming that that's ethically appropriate. And sometimes it's not. There may be sufficient, background data suggesting there's just no way that patients who are negative are going to benefit from this agent. But if there's, reasonable equipoise, this is the way to go because it gives you the most information. I have to tell you, though, it's very hard to enroll patients to a trial like this because they just up front, if you tell me you're going to randomize me to an EGFR targeted agent and I don't have EGFR mutation, that doesn't real attractive to me, okay? And it doesn't sound real attractive to clinicians either. But, we've had some surprises. Sometimes what we think is the right marker turns out not to be the right marker, and there are off-target effects. For example, in HER2, we are spending lots of money to do a whole new trial, trastuzumab, where we take the HER2 lows, the ones who would not have gone on those original trials, to figure out if, in fact, there's benefit of trastuzumab in those HER2 low patients. Jeff Oxnard earlier today touched on this. There are questions we can ask. I sort of talked so far about, give me the new marker and could show that there's benefit in using the new marker. But what if we have an alternative marker? So this issue of can we use cell-free DNA instead of a tumor biopsy to evaluate EGFR or any other marker, is that just as good or possibly even better way to select patients? This is a really tough problem, and Jeff alluded to this already, that you have to think about what you know and what you're trying to figure out. So basically you have 4 possible outcomes for the testing if you were to run both tests on all patients. And we know from our historical data that roughly a certain percent will be positive for the marker. We know, say, a response rate for those patients who are positive on that target agent. So if life were kind to us, we could say, well, let's look at the cell-free DNA assay for, again, let's suppose it's EGFR mutation. And if we could show that we pick up a big portion of patients, in other words, the prevalence in the plasma was actually higher than in the tumor tissue, and we could maintain the response rate in that group, that would be a success, right? But things don't always work out like that. So suppose you get a result like you've increased the pool of patients who are positive, and you don't quite match the response rate that you got when you had the smaller group defined by tumor biopsy. So how do you interpret that, right? Well, is that because you've diluted the group of patients who would benefit, that it was really only the ones who were too positive that would have benefit? Or does it mean that that extra group you've now picked up, the ones who are positive in plasma and negative on tissue, just biologically different, and they're still getting some response, but it's not high as the ones that are exclusively tumor positive. So how do you interpret that? And it takes a huge amount of data, a big trial, to really sort this out. So I don't have any good answers at this point. I would only say that there's yet another way you could look at this problem, which is the sequence of testing, and you heard about that. So suppose you say, well, all right, I'm going to test the plasma. If it's positive, I'm done. I'm not going to subject the patient to the tumor biopsy. If they're negative, then I'm going to follow up if I can find something in the tumor. Again, the way you're going to evaluate that is very tricky because you're now mixing together 3 groups of patients. The one you're not putting in the mix are the ones that were negative on both assays. So you don't really know how to interpret that response rate that you get out. Here's where, a randomization could be useful. You take those different subgroups and you randomize and you say, do these people actually benefit or not from the new targeted therapy? I think I'm probably just about out of time because we're running behind anyway. Some similar considerations apply when you're using biomarkers for monitoring, either after treatment, during treatment, or after treatment. You know, there are issues of confusing prognostic and predictive. You have to also think about when's the best time to measure the biomarker and the clinical actions you're going to take. And so I'm just going to speed ahead here to the punchline. There was actually a trial done looking at circulating cells, so not cell-free DNA, but the actual tumor cells, and they were following patients with breast cancer, and they said that if after 21 days of therapy you don't see those circulating tumor cell counts going down, that means probably those patients are not benefiting much from that therapy. The way to figure out if this is actually clinically useful information is to randomize those patients. Say if you switch the therapy, do you actually give them a better outcome? Well, the unfortunate answer is shown in the bottom right plot, that no, in fact, that this information did not turn out to be clinically useful because switching the therapy made no difference in their ultimate outcome. Now, what is that telling us, however? There might be that the treatment that they switched them to is just not the right treatment to switch them to. Now, maybe some molecular characterization of those CTCs would have given you some better information to tailor this alternate therapy. Maybe we weren't measuring CTCs at the right time. Maybe we should have done it earlier or later. And again, maybe some molecular characterization would have been helpful. Okay, so to sum up, I think it's important to identify the clinical setting and define the clinical question as early as you can in the process. Sometimes we get hung up, show lots of correlations without thinking about where we're going. And so also it's important to realize that the clinical utility of a biomarker test will be intimately tied with treatments you have available to actually take action, and treatments have to be effective when you take that action. And so we have to be keeping track of all these kinds of validation, analytical, clinical, and clinical utility. So planning, one of the difficulties we've had in this field of cell-free DNA assays is we have not traditionally collected a lot of blood samples in our old trials. Now we have to do that, but do we have ways to collect them that they're really going to be preserved and useful for the right analyses? Thank you.
Thanks, Dr. Shane. Or we collected the